Welcome to the central nervous system. Here we focus on the brain and the spinal cord. Your brain weighs about 1,600 grams in men, 1,450 or 1,450 in women. There's essentially an equal brain weight to body weight ratio between men and women, which accounts for that weight difference. And then we have our spinal cord, which is really our main line of communication between the brain and the rest of the body. So we're going to dive in and take a look at all things central nervous system. And there's a lot in this, so take your time, stop and replay as needed. So there are four items used to protect our central nervous system. Of course, we have bones, such as the cranium and the vertebrae, so all those bones that you studied in the skeletal system. And then we have our vertebrae, through which you've seen how the spinal cord passed. We have our meninges, which are membranes between bone and nervous tissue. They're the protective covering of the central nervous system. We have three meningeal layers. Kind of not surprised, right? We have this rule of threes. So we have three layers of connective tissue. We have our strongest, thickest outer layer known as the tough mother or the dura mater. And it's two layers around the brain. We have the periosteal layer, which is the inner surface of the skull. And then you have a meningeal layer, which is the outermost covering of the brain itself. In the middle, we have our arachna mater or arachnoid mater, the spider mother. This is a cobweb-like subarachnoid space filled with cerebrospinal fluid and blood vessels. And then we have our pia mater, the tender mother. And this one's known as the tender mother because it's actually clinging to the brain and to the spinal cord itself. We have our CSF, our cerebrospinal fluid, which is there for protection. It's produced by the choroid plexus or the plexuses. This is our kind of our waste removal, and then it's kind of buoyancy to spread the impact of force from kind of the brain crashing into the bone itself or protect from the blow. It's going to lighten the brain by about 97% because it's essentially floating. And we have this, it's like a watery fluid cushion and circulates through the ventricles in the brain and then drains back into the blood. Okay? Again, it's all about buoyancy. And then last but not least, we have this selectively permeable blood-brain barrier. And it's responsible for maintaining a stable brain environment. Blood's actually toxic to the brain. So this barrier is going to allow passage of water, glucose, and essential amino acids while preventing the passage of toxic waste. But it's useless against fat, gas, and fat-soluble molecules, things like alcohol, nicotine, anesthetics, and more. So those are your four means of protection of the central nervous system. So we have two types of central nervous system tissues. We have our gray matter, which are outer areas that are nerve cell bodies known as nuclei, dendrites, and unmyelinated axons, areas like your cerebral cortex. And then we have our white matter, which are deep fiber tracts. These have myelinated axons, or dense collections of myelinated fibers, and they have neuroglia. And an example here would be your corpus callosum. Your nerves originate in two areas. Your cranial nerves, arise in the brain, and your spinal nerves, spinal for a reason because they arise in the spinal cord. This is a great view, anterior and left lateral views of your ventricles. Remember, we were talking about how your watery fluid, your cerebrospinal fluid, circulates through the ventricles in the brain and then drains back into the blood. So we have hollow chambers here that are filled with cerebrospinal fluid. Again, created by the choroid plexus. So you have a pair of lateral ventricles. That's the first two, one within each cerebral hemisphere. And there's a communication channel between them known as the interventricular foramen, or the foramen of Monroe, and that's how they communicate. The lateral ventricles will drain into the third ventricle, which is in the diencephalon. And here you have what's known as a cerebral aqueduct, which is creates that continuous pathway, third ventricle continuous with the fourth ventricle via this aqueduct running through the midbrain. And then you arrive at that fourth ventricle, quite small, honestly, and it's kind of dorsal to the pons and superior to the medulla oblongata there, and it connects with the subarachnoid space of the brain and is continuous with the central canal of the spinal cord. So they drain continuously and are connected together. So a pair of lateral, and then the third and fourth ventricles. So 4 ventricles, or 4 hollow chambers filled with cerebrospinal fluid. So this is just a lateral view of the general regions of the brain. We're going to take a look at all of these. The main area there, the cerebral hemisphere. Sandwiched between the two halves would be your diencephalon, and then your brainstem, which is broken into 3 parts. And you have the cerebellum at the back, or the posterior portion of the brain. And again, we're going to get to know these quite well, but this is just a general view of the lateral view of the brain. And this is just here as another way for you to start memorizing all sorts of really good stuff. It is a mid-sagittal section, or a cross-sectional view down the center, and it labels all sorts of really lovely things here, and you can see where you find those ventricles, where you see the midbrain, where that cerebral aqueduct is, and then kind of all these general structures that we're going to talk about. And it's just a lot of memorization, knowing where things are. But there's some really neat things in the brain, so getting to know this is kind of fun. So we start with the cerebrum. This is 80% of the brain mass and deals with your higher mental functions. You have ridges, shallow grooves, and deep grooves. And those are known as gyri, sulci, and fissures. We're going to take another look at these again, so you'll see another picture and some more information. But you get your cerebrum divided into right and left halves. They're divided by what's known as a longitudinal cerebral fissure and are connected internally by the corpus callosum, which is a white matter tract. So review white matter. Each hemisphere is going to control the function of the opposite body side, and this is known as contralateral control. Make sure you understand that. Okay. Contralateral control, again, is how the right hemisphere will control the left side of the body, right, and vice versa. So each hemisphere is going to control the function of the opposite body side. And again, make sure you understand that they communicate with each other via the corpus callosum. That's that white matter. We have kind of what's known as lateralization of function. And that's going to be where your right hemisphere has more to do with kind of insightful matters, spatial, artistic, intuition. And your left side is going to be more language, math, logic, so things like analytical and verbal ability, and usually right-handed, right? That's typical. You can see that we have 4 lobes, and I've broken those down for you with a little bit more detail as to what's going on. And we will see a little bit more about this when we talk about some of your specialty areas, motor and sensory areas, along with some of the association areas. So we'll get some more talk, but this is kind of your general what's in what lobe, what controls are in what lobe. So this is a nice superior view, and you've got the anterior and posterior labeled on there. You can see that rather large longitudinal cerebral fissure. So again, fissure being deep groove. You can see where you have your frontal lobe, parietal lobes. Remember, they're going to be on both their paired parietal. So frontal, both on either side of that longitudinal fissure, paired parietal, right, and occipital lobes. And this gives you a little bit of a view of the veins and arteries covered by that arachnoid mater and what that looks like before it's kind of pulled away. And it tells you which is your right versus your left. So hold up your hands to my screen real quick, right? And because you're looking at the screen, you've got right on the right-hand side, left on the left side. Oftentimes when you have a specimen on the table in front of you, you have to cross those hands over because the right is on your left and the left is on your right. But in this case, I've got it set just for you, so you're looking left to left and right to right. This is a lateral view instead of that nice superior view. You can see your central sulcus, so that's a shallow groove, but it's quite important, and you'll learn a little bit more about that later. Then you have a precentral, meaning toward the front of the central gyri, so ridges toward the front of the central sulci. And then you have a postcentral, which means toward the posterior side of that central sulcus. And those are going to hold some pretty special areas, so making sure to kind of have an idea of where they are. So you have that one shallow groove, and you have an important section immediately toward the frontal lobe and one in the parietal lobe. And then it gives you some more of those fissures and sulcus, and you can still see your cerebellum pons, medulla oblongata, and how it's continuous with the spinal cord. So we have some layers of the cerebrum here. You have your cerebral cortex, which has an outer layer of gray matter, billions of neurons, and approximately 40% of the brain mass. This is considered your conscious mind. And you have things known as convolutions, which are the gyri and sulci within the cortex. Then we also have our white matter, which is your inner layer, and these are your connections within the brain. So these form connections via three types of fiber tracts. You have your association fibers, which are going to be confined to one hemisphere and conduct impulses between neurons just in that one hemisphere. You have commissural fibers, which are going to connect neurons between the hemispheres, such as your corpus callosum. And then we have projection fibers, which have descending and ascending tracts, which means they're going to have information and they're going to travel between the brain and the spinal cord, things like corona radiata. Okay, so those are your three types of fibers. And then we have our basal nuclei, which are masses of gray matter deep within that white matter, and they're associated with voluntary and involuntary activity. So they're going to kind of play a role in the intensity, the speed, the inhibition of antagonists. They also play a role with cognition and emotion, and they help to filter some responses. So those are kind of our layers within that cerebrum. Right? So cerebral cortex, the outer layer, white matter, the inner layer, basal nuclei, gray matter inside that internal white matter. This is just your visual representation of those fiber tracts in the white matter. So you can see where it's labeling your association fibers, your commissural fibers, and your projection fibers, right? And so you can see with the association fibers, which they have there on the visual A, they're in red, and you'll notice that they stay only on that one hemisphere. You have your commissural fibers, which are going to cross. And then you have those projection fibers, which you can see running all the way down into the, past the medulla oblongata and into the spinal cord. So this is kind of what we were just talking about. So Brodmann areas refers to the structural and functional map of the cortex. We have motor, sensory, and association areas. And this slide is super, super general. There's a lot to learn here. I'm going to try to run through some things to pay attention to. But I think your handout goes into more detail. So my notes template, which will be super helpful to you, but I'll point out a few things and maybe give some hint, hint, nudge, nudge, wink, wink as we go. So we'll start with our motor areas. This is the posterior frontal lobe. This deals with voluntary movement. And you can see that we've got 4 motor areas. So you have your primary motor cortex, and here's that hint, hint, nudge, nudge. It's located in the precentral gyrus, making sure you understand that that's in the frontal lobe section in front of that central sulci. This is your precision, so voluntary movement. Your premotor cortex is anterior to the precentral gyrus. Okay, so it's in front of that precentral gyrus. This is learned motor skill, and this is going to be more coordinated movement. You have Broca's area, which is your motor speech area, and it's only typically found in the left hemisphere. This is active as you prepare to speak. So if you have Broca's aphasia, meaning damage to the Broca's area, your damage might stop you from being able to articulate what you want to say. And then you have your frontal eye field, which is voluntary eye movement. It doesn't have anything to do with vision, just eye movement. We have our sensory areas next, and that is dealing with conscious awareness. And you'll see that it's located in all of those 3 lobes. The primary somatosensory cortex is in the post-central gyrus of the parietal lobe. This is receiving information from skin, skeletal muscle, and joints. This is contralateral specifically. And then there's spatial discrimination. Somatosensory association cortex integrates different sensory inputs, things like temperature, pressure, or from your primary somatosensory cortex, right? So it might integrate what you're getting from your PSC. And then it's going to relay to primary areas. Your visual areas are going to receive visual information from the retina. That one's at the extreme posterior tip of the occipital lobe. And within this, you have your visual association area. So you're going to get the interpretation of your visual stimuli. You're going to use your past experience, visual memory, facial recognition, right? All of that is located with the visual areas. You get your auditory area, which is located in the temporal lobe. Pitch, rhythm, loudness, location. The auditory association is have that perception of sound stimulus, so quote, hearing. The olfactory cortex, this is both frontal and temporal lobe. This is the awareness of different odors. Odors. Your gustatory cortex is in the parietal lobe, dealing with taste. Your vestibular cortex is deep to the temporal lobe, dealing with balance. This is more really when I talk about balance with this cortex, I'm talking about the position of the head in space. And you have a visceral sensory area, which is the perception of organ sensations, such as like a stomach ache. Then we have our multimodal association areas. This is going to be the integration and processing of information, communication within the brain between various primary and association areas. It really, what it's going to do is give meaning to the information received, store it as memory, compare it to previous experiences, and decide on action. So we have our prefrontal cortex. This is your higher levels of hearing and seeing, so to speak. This is kind of also known as your anterior association area. This is the most complicated. Prefrontal cortex has a lot going on. And it's kind of the last to rewire when the brain goes through rewiring through kind of that adolescent or daredevil type ages. This is the frontal lobe, intellect and complex learning, reasoning and judgment and conscious concern for others, personality, working memory for persistence, conscience, some of those things. The posterior association area is temporal, parietal, occipital lobes, patterns, faces, localization, and space. Then we have our language areas, left hemisphere only, comprehension and articulation. So the ability to understand and articulate come from this area, this association area. You have your Wernicke's area, which is your primary language area. This is the ability to understand written and spoken language. And it's kind of this body language piece, right? So understanding. If you have aphasia or damage to the Wernicke's area, you'll have the inability to understand language. So you might be able to speak, but it potentially will be unintelligible. Very interesting piece of this language area. Next up, we have general interpretation area. This is kind of really not well defined at this point. And it's across multiple lobes, and it kind of takes in a lot of incoming signals and translates to one output. So general interpretation of information. And then you have your visceral association area, which is that consciousness of those visceral sensations. So we talked about the visceral sensory area in the sensory areas a little bit, which is that perception of the organ sensation. And then you give conscious reasoning to what that means in the association area. So a lot. I would kind of go back and look at this. There's lots of information in the book, but keep it simple, understand where it is and the general purpose of it. There were some hints in there, right? So, well, I definitely gave one hint in the motor area for sure, right? Your premotor cortex. And then you've got some understanding of the prefrontal cortex and the Wernicke's area, right? So kind of keeping that all in mind as you move through this. And we're going to take a look at a couple pictures here as well before we move on, so I can kind of point out some more stuff and things. That's very scientific. So here is your first diagram of the Brodmann areas. You can see your motor areas, your prefrontal cortex specifically, yay, and what's going on with it. And then your sensory areas and association areas. You've got a nice label, your diagram, your color code down here at the bottom. So you have your primary motor cortex, your motor areas, sensory areas, and the association cortex. Okay, so the red and the light red are dealing with motor. There's your central sulci located for you. Then you have your sensory areas in the shades of blue. And then you have your association cortex in kind of this purplish color. So it goes through all of these areas again that I just kind of walked us through on the previous slide. This is a nice way for you to kind of review and put kind of the words that I just spent too much time just blabbing at you about into your picture. And one more time, just because it's fun, except in cross-section. So now you can kind of get a different view of some of the different things that we were talking about, but from the cross-section view, right? And it gives you your corpus callosum, so you can see where those ventricles and your cross-communication comes in as well in relation to these different areas. So one more view for good measure for you. This is one of my favorites just because it's easier for me to see, but this gives you quick look at, just a brief look at some of the major regions, right? You can see your primary motor area, your somatic sensory area, the primary visual cortex, Wernicke's, Broca's, your auditory and olfactory areas, and it's just in a quick color code so you can see where in relation to those lobes they fall, and then the central sulcus is labeled for you as well. So just another view. I know, lots of views, but another view so you can kind of piece together where all of these Brodmann areas are located. All right, now that we survived the cerebrum, we can finally move into the diencephalon. The diencephalon sits on top of the brainstem and is enclosed by the two hemispheres, your two cerebral hemispheres. It also encloses the third ventricle. There are three major structures and two glands that we find as part of the diencephalon. The thalamus makes up about 80%. This is the relay station for sensory impulses and kind of gives us a hint as to whether the sensation is going to be pleasant or unpleasant. It's going to sort, edit, and relay the information. It's kind of the gateway to the cerebral cortex. You get some motor activity, cortical arousal, learning, and memory. The second part is hypothalamus, which is your primary link to the endocrine system. So it plays a pretty vital role in homeostasis. There are a lot of nuclei here, and it connects to the pituitary gland, which has endocrine function. So it also has some autonomic nervous system regulation, like your heart rate and blood pressure. You can see a lot of other things listed here on the screen. Gave you a bunch of that. And it produces the posterior pituitary hormones like ADH and oxytocin. So it does play a pretty key role with the endocrine system and the endocrine link. And then we have our epithalamus, which is going to contain your pineal gland. Your epithalamus has the choroid plexus. Here's where we produce our cerebrospinal fluid, and we produce melatonin for the sleep and wake cycle, known as your circadian rhythm. The diencephalon, three major structures and direct connection to autonomic nervous function and your endocrine system. And here's what that enlarged cross-section looks like. If we were getting to do any sort of dissections whatsoever, or showing you live tissues, the thalamus does actually kind of look like a button. And so you can, once you can find that button known as the thalamus, then you can travel slightly anterior and inferior. You find your hypothalamus. And because you can find the pineal gland, you know you're at the epithalamus. And it's always fun to do dissections and be able to do a cross-section of the brain that allows you to find the pineal gland and find the pituitary gland. Super fun, because that interthalamic adhesion is intermediate in that mass of the thalamus, which is super fun to kind of realize where you are and what you're seeing. So that's it, just a quick visual. Next, we find ourselves at the brainstem. This is going to connect the brain to the spinal cord, produce the rest of our automatic behaviors of survival, and gives origin to our cranial nerve. So acts as a pathway for ascending and descending tracts. Really interesting. If you hold up your thumb, give yourself big thumbs up. That's it. That's your brainstem. And it has so much going on. So as we talk about the four major parts, think about your thumb and the size of your thumb and what's going on with your brainstem. First up, we have our midbrain, which is the pathway for impulses, both visual and auditory reflexes. Houses the cerebral aqueduct, cranial nerves 3 and 4. We have a lot going on. So we have what's known as a superior colliculus. This is to coordinate head and eye movement for tracking, so for visual purposes. Inferior colliculus is your auditory relay. Substantia nigra functions with basal nuclei, so the release of dopamine. Subconscious muscle activity, so when you deal with Parkinson's, you get the loss of neurons here in this area. And then we have our red nuclei, which is that voluntary limb movement, so limb flexion. That's already a lot, right? Superior colliculus, head and eye movement, inferior colliculus, auditory, substantia nigra functions with the basal nuclei, red nuclei, motor pathways or voluntary limb movement. As we move inferior, we run into the pons, which is the control of breathing. You get cranial nerve nuclei for 5, 6, 7, 8-ish. Sometimes listed with the medulla oblongata. This is going to connect the motor cortex and the cerebellum and connect higher brain centers with the spinal cord. Then we have our medulla oblongata, everybody's favorite. Thank you, Adam Sandler. This is the control of heart rate, blood pressure, breathing, swallowing, vomiting, so much more. Potentially that cranial nerve 8, but definitely 9, 10, 11, and 12. We get ascending and descending tracts, so you get sensory information from muscle and joints to the cerebellum and responses to help maintain equilibrium. You have tracts that cross at this location known as cessation. And you have these really wonderful autonomic centers, which are all those things I was listing, right? So the cardiac center for accelerating and inhibitory fibers, right? So force and rate of the heart. Vasomotor for vasoconstriction, vasodilation. You have your rate and depth of breathing, and then all those other things that I talked about. And then we have our reticular formation, which plays that role in consciousness and a week's sleep cycles. So this is your state of alert consciousness. Okay? Hint, state of alert consciousness. You get your reticular activating system and motor function. So you get limb movement, visceral motor function, vasomotor, cardiac, respiratory, right? So this is kind of a network of nuclei and fibers throughout your medulla pons and your midbrain. And it's continuous impulse flow to the cerebral cortex in order to monitor sensory impulse. It's going to kind of filter out unimportant stuff, the background stuff, and really if you want to know what it filters out is like 99% of stimuli. Get a quick laugh out loud there. So the reticular formation is going to essentially filter out about 99% of what's considered background nonsense and help you really focus on your state of alert consciousness. If you get severe injury or if you have severe injury to the reticular formation, it can really result in permanent unconsciousness. So very interesting that the reticular formation is quite important. And I think that covers a lot. So again, hold up your thumb and think about the fact that I spent a really long time talking about this thing the size of your thumb. And there's a lot going on there. Are you still holding up your thumb? All right, well, take a look because here it is. Midbrain, pons, medulla oblongata, and inferior onto the spinal cord. Notice that the pons has a little bit of an extended kind of lump look to it. So that is how you know that you're at the pons. Midbrain will be superior, medulla oblongata inferior to that area. I think that pretty much covers it. As far as that part, we're going to take a couple more looks at things. I'm going to show you a couple more pictures here. This is a ventral view with everything kind of stripped away. The cerebral hemisphere is in the occipital lobe and the frontal lobe and the parietal lobe and everything gone except for this interior tract here. So you can see the diencephalon. You get a bunch of the nerves. This is important, so pay attention to that. We are going to go over them again. Don't worry. But you get to see the part of the midbrain, and then you get your pons. And then if you just pay attention, right, when you leave the pons, that kind of greenish area is the medulla oblongata, and it's continuous with the spinal cord. The midbrain, pons, medulla oblongata, and you can see where all of these lovely nerves find their origin, or at least 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. And you'll notice that 10 and 12 are like right on top of the thing. You get to know, you can start to know where those nerves arise as well. And last, but certainly not least, is a view of the reticular formation. Remember, we're talking about this state of alert consciousness and all the things that the reticular formation is going to pay attention to and essentially filter that unimportant stimuli. So you can see you get visual impulses and auditory impulses and radiations in the cerebral cortex. And somebody's got to pay attention to what's going on. You get sensory tract, motor projection, right? There's a lot. And so the reticular formation is going to sort out what needs to be paid attention to and what doesn't. So this is where that's kind of going to occur. And the last section before we move into the spinal cord is the cerebellum. This is only about 11%. It's found kind of inferior to the occipital lobe of the cerebrum. Looks like cauliflower. Not a nice way to put it, but we're going to coordinate skeletal muscle movement, contraction, recruit motor units, react to proprioceptors. There's a lot that it's going to do with cognitive function, recognizing and predicting event sequences, non-motor function like word association and puzzles. And if you lose it, you get some clumsiness. So we have cerebellar processing, which is frontal motor association area notifies the cerebellum of the intent to initiate voluntary activity, and it's going to say yay or nay, essentially. And it's going to kind of receive some of that worldly information, right? So you're going to get some automatic piloting, auto-correction going on. It's going to provide what allows body movements to be coordinated and smooth, and provide feedback, right? So kind of that blueprint, what are we doing? How are we going to do it? And how do we get the precision of timing and patterns? And then if you do a cross-section of it, which I'm going to show you on the next slide, you have what's known as the arbor vitae, which is the tree of life and is white matter. So keeping in mind important information about white matter. Quick view, lateral view, the pinkish looking cauliflower sort of thing hanging off the bottom of what you consider the brain, that's your cerebellum. That's it, nothing too special here, that's what it looks like, although it's kind of important for coordinating all of our movements and not being clumsy all the time. But that's what it looks like. And as promised, here's the cross section. You can see the arbor vitae, looks like a tree. So it's the tree of life. It's all of the white matter. So here's that for you. You can see that. And then you get a nice little bit of a reminder here of the pons, the fourth ventricle, the medulla oblongata, the choroid plexus, where we make CSF, and kind of this understanding that you're looking at it kind of cut in half, right? So that midsection, mid-sagittal view. So we talked about the reticular formation and the importance of the reticular formation earlier, right? That you're talking about the state of alert consciousness. We also have what's known as the limbic system, and this is the ultimate last thing dealing with the brain itself. Limbic system or limbic association area are groups of structures on the medial hemispheres. This is kind of the affect, right? So we had our reticular formation, which was dealing with alert consciousness, and now we have our affect or our affective brain. Emotional drives, the amygdala for eliciting the fear response. Here's where you can tie emotion to order, visceral response, interact with the frontal lobe to communicate your thoughts and emotions. So you have reasoning versus emotion going on. And both the hippocampus and the amygdala play a role in memory. And so you can see both of the amygdala and the hippocampus on the right-hand side there. And this is kind of just showing you the structures dealing with your limbic system. And again, this is our affective brain. Right, so I'm keeping that in mind. I don't have any hints for you here, though. And here we go. We're heading into the spinal cord, finally. So we leave the brainstem via the midbrain pons, medulla oblongata, and we are continuous with the spinal cord. Spinal cord has two functions: impulse coordination through the ascending and descending tracts, and reflex integration. Those not voluntarily initiated by the brain. Okay, so your spinal cord extends from the foramen magnum to L1, L2, lumbar. Just in case you need a reminder, down the lumbar. The terminal end of that is known as the conus medullaris, with a fibrous extension called the filum terminale to anchor the cord to the coccyx. Okay, so again, it does get anchored, so don't forget that it's not just kind of free floating in there. The cauda equina are nerve roots that extend inferiorly from the conus medullaris through the vertebral canal prior to exiting. You have a cervical enlargement for the area of where the upper limb nerve arises, and a lumbar enlargement for the lower limb nerve. And you get to see a lot of the same protective features. So you get your meninges, CSF, you get cushion of fat, network of veins. It is secured to the dura mater by the denticulate ligament. So that kind of interesting. And we're going to take a look at the growth structure and the anatomy of the spinal cord. When we talk about the ascending tracts, you get travel from the body to the brain. That's your sensory information. When we talk about descending tracts, that's travel from the brain to the body, right? That's motor information. And it kind of is how the lesions are present. So your upper motor dealing with your primary motor cortex, lower motor, probably ventral horn, and innervating your skeletal muscle. And then you do have a transverse tract, which is going to be kind of traveling to the opposite side of the cord. And then there are 31 pair of spinal nerves. 31 pair of spinal nerves. How many? 31. I bet I've repeated that for a reason. And then we're going to look at the cross-section in just a moment as well. So here is a quick picture of the growth structure of the spinal cord. You can see the cervical enlargement, the lumbar enlargement, right? And then your conus medullaris, cauda equina, and the filum terminale, so that it's anchored to your coccyx. So it's up there floating around. You don't have anything to see with the dura mater here, but just understanding you've got cervical spinal nerve, thoracic spinal nerve, lumbar spinal nerve. Hopefully you're thinking back to the skeletal system and how you had the structures and the different outlets for the nerves, and you can think about how your compressed vertebrae could cause a problem with the function of the spinal nerve. I just wanted to provide you a little bit of a cadaver type view, what we might see if we had been able to really look at it. Really interesting. You can see the end of the medulla oblongata, right, the brain stem, and it's pointed at, and that it's continuous with the spinal cord. You can see some spinal nerve rootlets, which is really, really awesome. You can see where they cut the cervical vertebrae away there. You can see the end of the pedicles, and you can actually right at the top see that dura mater. This is a nice dorsal view of the spinal cord. All right, this is an important anatomy of the spinal cord picture. I'm going to tell you that you'll see this on the lab test. So become familiar with what might look like something like this cross-sectional view here. You need to understand that you've got a dorsal and median sulcus there, so you understand what you're looking at from anterior, posterior, right? You have on this picture, you can see the dura mater, arachnoid mater, and the pia mater, and the difference in how they kind of are coverings that get closer and closer to the actual cord itself. You've got your gray matter, you've got white matter in the form of columns. White matter is going to be mostly ascending and descending sensory tracts for motor, and transverse tracts like commissural cross fibers, commissural crossing the sides across the way. Make sure you know your anterior versus posterior horns and the gray commissure. So you should be able to load that. Okay, this is your cross section. This is another picture I would become very familiar with. Okay, this is showing you the body of a vertebrae, so you can see the actual vertebral structure and how your meninges align in there. And it gives you kind of that view of the spinal cord. It also shows you where the space is for the CSF. This one not as much as the other one. I really would make sure that you can label all of the pieces from the lecture handout, cross-section view list, kind of know where all those things are, and making sure you understand anterior from posterior so that you know what you're identifying. And for sure, you're going to see this on the lab test. I wouldn't lie to you. I absolutely promise. Yeah. So one of the things I want to talk about real quick on this slide before I set you free has to do with spinal cord trauma. So paresthesis is sensory loss. Paralysis is motor loss. So making sure you understand it. Paresthesis, sensory. Paralysis, motor. Flaccid paralysis is loss of lower motor neuron. So no contraction, so no voluntary or involuntary. You're going to have a lot of atrophy. Spastic paralysis is loss of upper motor neuron, and it's just no voluntary control. So when you're dealing with spinal cord trauma, where you have the transection of the spinal cord, right, anywhere, at any level, the cross-sectioning at any level equals the motor and sensory loss inferior to that transection. So whatever point along your spine you can think of, wherever that gets cut, everything inferior of that is essentially lost. So paraplegia is motor and sensory loss of both lower limbs, because the transection is around T1, anywhere T1 to L1. Quadriplegia is the motor and sensory loss of all four limbs, so you have a transection at the cervical level. Hemiplegia is loss of one side of the body, and it's usually secondary to some sort of brain injury. And again, if you injure the left, you could have some of that hemiplegia on the right. Right? So paraplegia is dealing with thoracic and lumbar injury. Quadriplegia, all four limbs, that's cervical region. And hemiplegia, that's when you have that secondary outcome from typically a one-sided brain injury. So just kind of keeping that in mind, you don't necessarily need a whole lot of specifics there. All we're going to do now is just kind of take a quick peek, and I'm going to point out a table to you for review. So on this, these next two slides, we're going to go over the functions of the major brain regions. So on this first of two, we're going to talk about the cerebral hemispheres. You can see the cortical gray matter, the basal nuclei, and then the diencephalon, which is where we house the limbic system, but also, our coordination with the endocrine system. And last but not least, our brainstem and a cerebellum, so the three parts of the brainstem and all the amazing things that are going on there, along with the reticular formation, and then our cerebellum, and just that little thing that smooths out all of our rough edges. And that is everything. Remember, I already mentioned the lab test, and yes, it's a lot of muscle identification, which you should be practicing using both the Quizlets, hopefully they still work, as well as the Kahoots to practice. But then please take a look at the cross-section of the spinal cord so that you can understand the anatomy and all the parts, because that does absolutely show up on the lab. And all we have left now are peripheral and autonomic lectures. So we are almost done.